let's now enter inside a blood vessel and look at the blood flowing through it. And there are creeks and crevices where each and every component of the body is going to receive what it requires through these vessels. And now let's zero in on one by one every cellular component of the blood. Let's begin with erythrocyte. Now, as I mentioned, erythrocytes are also called as red blood cells. Now, looking at the structure of the red blood cell shown over here, it is biconcave in nature, so that though it is disc shaped, it's circular but compressed in between, and that compression, that biconcavity, actually helps in increasing the surface area for accommodating more of the hemoglobin. Now, there are certain things that we need to know about the red blood cells or the erythrocytes. First, of course, let's begin with the size. Now, the size of the erythrocyte will be, the length is anywhere between 7 to 9 mu m in diameter, in length, and 2 mu m in diameter. All right, that is the size. Now, the red blood cells are maximum in number in the blood, and because of that red blood cells we get the characteristic red color. So what is the number of cells, RBCs, will be, we have almost 5 million RBCs per 1 cubic mm of blood. That is the number we are looking for. Imagine 1 cubic mm of blood, it's hardly even a drop. In that we have 5 million RBCs. It's average, on an average it is 5 million RBCs. Now, why is it so? Simply because RBCs are the one which are going to take the load of carrying oxygen and bringing back the carbon dioxide from the body to the lungs. Oxygen requirement of a living cell is of utmost importance if you compare it with the other requirements. You can manage without water, you can manage without food, you can definitely manage without hormones or other components, but you cannot manage without oxygen. And when we were doing respiration chapter, if you go back to the lecture of requirement of oxygen, I had mentioned about the irreversible damage that the brain undergoes due to lack of oxygen, even culminating to death. It is fatal. So, to accommodate the demand, to give the supply, we need this much amount of RBCs in that one mere drop of blood. Now, the lifespan of RBC, what is the average lifespan of RBC? It is about 120 days. Now, 120 days, when I say, it is not that after 120 days all the RBCs are dead. Does it happen that way? No. We say the average lifespan of human beings is what? About, say, 80 years of age. Does that mean after every 8 years the earth is devoid of human beings? No, because we have human beings being born every day and human beings dying every day. So at every point of time you will have the human beings of different age group, correct? Similarly, there are RBCs of different ages. Some might be 2 days old, some might be 10 days old, some might be 110 days old. So, like that, the average lifespan of one RBC is about 120 days. Now there's no nucleus or other cell organelles in RBCs. Now, why is that so? It has to accommodate hemoglobin, correct? So that because of hemoglobin, because it requires to load as many as possible hemoglobin in one single RBC, the nature has programmed it in such a way that when a RBC is born, when an erythrocyte is born, it has got nucleus. As it matures, it becomes enucleated or devoid of nucleus. And if there is mitochondria inside the RBC, the oxygen will be used up by that mitochondria for oxidation, which it cannot allow because it is only supposed to carry the oxygen. So it is like, if a sweet meat shop owner eats up all his sweets, what is he going to sell? Just like that, the RBC cannot afford to consume the oxygen for itself. In fact, RBC, it is such an ironical situation. RBC undergoes anaerobic respiration for its survival and supplies oxygen to the entire body. Then it has got hemoglobin. It consists of hemoglobin, which gives its characteristic red color to the blood. Now, heme, it is, as I mentioned earlier also, it is a component of iron as well as globin part. It is called, it is making up the pigment, the respiratory pigment of the blood, that is hemoglobin. Then, if we look at the formation of red blood cells or erythrocytes, it happens in the bone marrow. So, the formation of RBCs, or in other terms it is also called as erythropoiesis. It simply means formation of RBCs. It happens in bone marrow. Do you know where it happens in the fetus? The fetal blood is formed in the liver, but in case of the and as the baby is delivered, the erythropoiesis or formation of RBCs is taken over by the long bones, and that is the bone marrow of the long bones will take up this work. Now, when there is excess or abnormal growth of erythrocytes or RBCs, that condition, it is called as polycythemia. Now, polycythemia is where when there is excess formation of RBCs, and it is the opposite thing, that is the reduction or reduced number of erythrocytes gives rise to a condition called as erythrocytopenia. Now, this word is actually taken from the word penury. Penury is poverty. All right. So when there is dearth of RBCs, when there is a tremendous reduction in the number of RBCs, it is called as erythrocytopenia, and the excess number, the abnormal number of RBCs is in polycythemia. Now, you know, when polycythemia usually occurs, of course, there are physiological disorders, but in higher altitudes, many a times when people from the lower levels, a lower altitude go, they are not able to cater to the oxygen demand because of the low atmospheric partial pressure of oxygen. And as a result of that, it is the way the body combats the lack of oxygen, and they will bring into circulation even immature RBCs. And that is the time when it is described as polycythemia. Whereas erythrocytopenia is obviously lack of RBCs that can happen due to dietary problems. The people are not taking enough of iron, they are anemic. All those conditions can lead to erythrocytopenia, where the number of erythrocytes is reduced. Now, that is all about erythrocytes we can write here. What we also need to know more importantly is the lack of nucleus in the matured RBCs. So there is no nucleus or other cell organelles in mature RBCs. That is what you need to also remember, because immature RBCs are definitely having a nucleus. Then let us move on to the white blood cells or leukocytes. Now you can look at the diagram of leukocytes here on your side. You can look at the leukocytes. You can see that there are different images of different types of leukocytes. Now let us pick up the first one, that is the neutrophils. Now neutrophils, they belong to the category of granulocytes, right? So there is the cytoplasm is typically granular, and look at the lobes of the nucleus. Now here all the WBCs are nucleated. They have prominent nucleus, and that is very distinct from the RBCs. So looking at the lobes of the nucleus, can you see that the neutrophils are having almost 5 to 7 lobed nucleus? Then you can see that why we call them as neutrophils, because they cannot be stained by acidic or basic stains, cannot be stained by acidic or basic stains, but they need neutral stains to dye their nucleus. Then they are forming almost 60 to 62% of the total number of WBCs that are in circulation. So they form about 62% of the total WBCs that are there in the circulation. And if you look at the structure carefully, the cytoplasm, as I mentioned, it is typically granular, and they are phagocytic in function. Phagocytic in function. Now what is phagocytic? Phagocytic is when the cells are behaving like amoeba. What does amoeba do? When it eats, it engulfs the food particle. Similarly, the WBCs, in fact, generally all the WBCs are the soldiers of our body. So whenever they come across any of this microorganisms or any alien body, they will engulf the food or engulf the microorganism and destroy it with the help of the lysozymes, and so they are phagocytic in nature. Now, moving on to the next, we can see the next image here, that is second one is eosinophils. Now eosinophils, they will have, look at the shape of the nucleus here, the eosinophils are having a bilobed nucleus. Can you look at the image? Then they can be stained by acidic dye like eosin, stained by acidic dye like eosin. Then the next point, they are almost forming about 2 to 3% of the total WBCs, correct? And what is their role that they are doing? They are slightly phagocytic. Now, why slightly phagocytic? Because they are doing much more important work apart from phagocytic. So, it usually happens that if required, there is if there are not enough other neutrophils around, and if it can be, need based, it will be phagocytic, but it does the role of detoxification. So this important work is done by the eosinophils. Then the next, the third one in granulocytes is basophils. Now looking at the structure of the basophils, again the nucleus, you can see that it is almost 2 to 3 lobed structure. All right. Then it will be forming about 0.5% to 1% of the total amount of WBCs in the body. And it plays a role in inflammatory response to particular injuries or inflammatory reactions, you can say. It is involved in that. Now, when we talk about the lifespan of WBCs, that is the leukocytes, they vary from few hours to even many years. Now, why is that so? Because their role that is involved in protecting the body against the pathogens, sometimes it will be just produced for that particular pathogen, and once the pathogens are killed, they are destroyed, self-destroyed, few hours. Sometimes, especially when we undergo the vaccination program, so what happens in vaccination? What do we exactly do? We introduce the attenuated or partly killed microorganisms which are rendered harmless in the body. Now, the presence of that microorganism will elicit the WBC's response. The soldiers are actually, they are waken up to fight with that microorganism, and those WBCs which are trained to kill that microorganism which has been purposely introduced in the body, they will remain in the circulation for many, many years. And that is how we are protected from that particular disease. Say I am vaccinated against polio. So when polio vaccine is being given to me, that means I am being introduced, the virus that causes polio is introduced in my body, which is going to elicit the response of WBCs, and they will be in the circulation for many years, right? And then if ever the polio virus actually enters, the harmful one, the soldiers are already prepared. They know their enemy, they know the style and the way the enemy is going to attack, and before the enemy can actually strike, the trained soldiers are going to kill them. And so those WBCs, those leukocytes will be there in the circulation for a very long time. So that is why when we say the lifespan of the WBC or leukocytes, we cannot actually number it. It ranges from few hours to many years. And it is, as I mentioned, it is need based. Now let's look at the agranulocytes. Look at the image carefully. You can see that here, look at the image. It is of two types, right? We have the lymphocyte. The first type is lymphocyte. Here, the nucleus is covering almost the entire WBC, right? Entire cell. So, nucleus is rounded and covers the entire cell. Correct? Now, lymphocytes are very, very important from immunity point of view. You can give them the name of the special commandos. They are extremely vigilant and they are always on their toes, alert, roaming throughout the body to check whether we are safe and sound. Now, lymphocytes, you will see that they are of two types. One is T lymphocyte and the other one is B lymphocyte. Now, what is the difference between them? Well, T lymphocyte is the one. Now, both the lymphocytes are also formed in the bone marrow, correct? But the maturation of the cells, that is the lymphocytes, the maturation happens in the bone marrow itself, that is B lymphocyte. And this lymphocyte goes to a particular organ called as thymus. So, there the lymphocytes mature and that is why they are called as T lymphocyte. Now, when we talk about their specific function, we know that the T lymphocytes, they are definitely, as I mentioned, they are the crucial soldiers or the commandos, the special commandos. So, they are involved in the cell mediated kind of response, that is wherever there is the cell initiates the reaction, the lymphocytes are going to react in that way. So, it is called as the cell mediated response, and the B lymphocytes are taking care of the general immune response of the body. Now, going to the next type of lymphocyte, agranulocyte, that is we have the monocytes. Now, monocytes will have a kidney shaped, look at the diagram here. The kidney shaped nucleus, which is very, very typical of the monocyte, and they are majorly phagocytic in function. So, they go around taking care of the different pathogens. They are not allowed to multiply and they engulf, their major role is phagocytic. So, this is how they take care, the lymphocytes and the monocytes take care of the body. In WBCs, there is something called as, there is a particular term that you need to know, and that is diapedesis. What is diapedesis? Look at the process happening over here, diapedesis. You can see that the WBCs are squeezing out of the blood capillaries. And why is that? Because they have spotted a pathogen and they are going to come out of the blood capillaries and engulf the pathogen. So, they are that alert, and that process of the squeezing out, we can write it down here. The process of squeezing out, the process of squeezing out of squeezing of the WBCs out of the blood capillaries it is called as diapedesis. Now, many a times in your exam, they can give you the image of diapedesis happening and you are expected to explain what the process is going on. And what is the process? That the WBCs are going to come out of the blood capillaries. So, imagine the blood capillary, the endothelium that makes up the blood capillaries has that much enough gap through out of which the WBCs can squeeze out and kill the pathogen by engulfing it. It is a typical amoeboid movement that we can see here. And now we can see the disorders related to leukocytes. We'll note it down here. Just as we have the excess number of RBCs produced, we also have a similar problem here in the leukocytes, and that is when we say that excess amount of WBCs are produced, it is the situation or the disease that is caused is leukemia. And exactly the way there is reduced number of RBCs, we have the reduced number of WBCs, and that is called as leukocytopenia. It's easier for you to remember because we have studied erythrocytopenia, leukocytopenia. Now these are the disorders usually we come across. Now leukemia, we all know it is a type of blood cancer where there is uncontrolled growth of WBCs. But polycythemia is not a type of cancer. It is different. Here the RBCs are increasing in number. Here there is abnormal growth or abnormal increase in the WBCs. Now moving on to the third cellular component, and that is the thrombocytes, or they are also called as blood platelets. Well, what are thrombocytes? Now, they are tiny broken parts of a larger cell, and that is they are the broken fragments of cells that are called as megakaryocytes. These megakaryocytes again are produced in the bone marrow and they are broken. Now you can see the structure of the blood platelets or the thrombocytes here, and they are just, you can see that they are just broken fragments, the components of bigger cells called megakaryocytes, which are produced in the bone marrow. Now their lifespan, since they are broken cell components, we do not have any nucleus or anything in that. Now their lifespan is about a week, and the amount, that the number of thrombocytes that we should have in 1 cubic mm of blood is about anywhere between 1,50,000 to 4,50,000 per cubic mm. That is the number required, and we are all aware that in diseases like dengue, where the virus attacks the platelets and the platelet level drastically comes down and the person can actually bleed to death. Now what is the role of thrombocytes? They have something called as thromboplastin, which when released at the site of injury leads to the coagulation or clotting of blood. Alright, it is this thromboplastin which triggers the series of events, and with the help of fibrinogen. Well, what was fibrinogen? It was the protein component, right, of the blood. Now they will set off the chain of reactions where the fibers are being formed and the cellular components get trapped in that fibers. We will be doing blood clotting in details later, but for now you understand that role is carried out by thromboplastin. And what is the average clotting time in healthy human beings? It is around 3 to 4 minutes, the normal clotting time. The normal clotting time. Alright. Now again we have thrombocytopenia, where there is lack of or a reduced number of the thrombocytes, and that can cause internal bleeding. Even that happens in the case of dengue. The internal bleeding takes place and the person can actually die because of this internal bleeding, and that happens when the platelet number drastically comes down. Now here the amount of RBCs normally seen in 1 cubic mm is 5 million RBCs. Here it is 1,50,000 to 4,50,000 per 1 cubic mm, and the WBCs range from, the range of WBCs again very high. We need around 5,000 to say 9,000 on an average, because there are different measuring units, there are different measuring instruments which give, some give 4.8, some give 5. So it is on an average, it is 5 to 9,000 WBCs per cubic mm. Now that is the amount required for a healthy person. That many numbers of WBC should be always there in circulation. Now this takes care of the cellular component. Next we need to proceed towards is the functions of blood. Now what role has the blood to play in our body?